Hello, my name is Juan Flores. I am a PhD candidate in the Molecular, Cellular, and Integrative Physiology graduate group at UC Davis. I work in Dr. Karen Zito's lab at the Center for Neuroscience. In our lab, we're interested in understanding the molecular and cellular mechanisms that underlie learning in the brain. To this end, we study the pyramidal neurons in the hippocampus, focusing on individual neuronal connections known as synapses. Most of the excitatory synaptic connections in the hippocampus occur at small membranous protrusions from the neuron's dendrites known as dendritic spines. My project looks at the cellular and molecular basis for spaced learning. Spaced learning is a paradigm of learning that incorporates breaks into learning sessions. This has been shown to improve the efficacy of learning and improves learning in animal models of Fragile X and Down syndrome. This begs the question, why is spaced learning more effective? One observation from Kristen Harris's lab is that plasticity can be saturated in the hippocampus and that saturation is released over time. My hypothesis is that saturation of LTP occurs at individual synapses. To look at this question, I am looking at cells in cultured hippocampal slices. I use bioelectric transfection and 2-photon microscopy to image individual neurons. Because we are using 2-photon excitation, we're able to excite only the fluorophores in the focal volume of the microscope. This 2-photon effect also allows us to stimulate individual dendritic spines with glutamate uncaging. The approach is simple. I find the spine and I stimulate it with high-frequency glutamate uncaging. Then we evaluate the volume changes as a measure of plasticity. I then stimulate that same spine 30 minutes later, and at this point, I stimulate a size-match control spine elsewhere on that same cell. We expect that the control spine will grow in response to the stimulus, so the question becomes, what happens to the previously stimulated spine? Here I'm showing some representative images that show the target spine before and after each of the two stimuli, and at the bottom we have the control spine. If we look at the volume graphs, we see that the spine has sustained growth after the first stimulus, but not the second. If we renormalize the second stimulus and plot it with the control spine, we see that the target spine does not grow like the control spine, indicating that its plasticity has been saturated. I next looked at the timing of the saturation I have observed. In this experiment, I simply increased the interval between stimuli from 30 minutes to 60. Here we see representative images showing the target and control spines before and after each stimulus. This plot shows that the increase in volume in response to the initial stimulus persists for 60 minutes, and that there is an increase in volume in response to the second stimulus. Plotting the second stimulus and the control spine on the same graph shows that they are nearly identical, indicating that the target spine has been released from saturation. Feel free to reach out to me with questions and come to my poster where I will talk at more depth about these results and more. And with that, I would like to thank the current and former members of the Zito Lab that have helped me throughout the years, as well as my funding from the NIH and the ARCS Foundation. Thank you.